Viruses are invisible invaders. They are too small to see, yet they are powerful enough to shut down an entire system, such as the immune system. But here is the question. Are viruses actually alive or they are not? Now they can replicate, they can evolve, they can cause disease, but they can't grow. They can't make their own energy or food, and they can't survive without a human host. So what exactly are they? How can something which is very small and we can't really tell if it's alive or not, something very small, how is it able to cause even global pandemic like the COVID-19, like the seasonal influenza or the flu, like the common cold, or like HIV and AIDS? So in today's lecture, we're going to break down viruses and prions. We'll be looking at their structure, how they replicate, how they cause disease. This is chapter 7 of our microbiology topic, viruses and prions, where we will dive into the detailed information about viruses. And we'll begin to get to understand the gray area between whether they are alive or not. How do they function within a host system? How do they multiply? And how do we even treat viral diseases and infection? We'll also look at some non-viral, non-infectious particles called prions, and we'll also look at viroids in this particular lecture. Now, if you take virus as a microbe, they are really abundant. So just like we've talked about for bacteria, viruses are also abundant, and they can infect every cell type that you can imagine. They can infect bacteria, algae, fungi, plants, animals, and viruses can even infect other viruses if you look at it in detail. But the big question is, are viruses alive or they are not? Now, when you think about microbes in the context of living things have some basic characteristics that allows us to classify them differently from non-living things. Now, can we consider virus among the living? Are they alive or they are not? And here is how we can put it. We cannot really classify viruses as being alive or not, because in terms of being alive, viruses do have genetic material, which living things also have. Viruses are able to evolve. Viruses can replicate and multiply themselves only inside a viable host. But on the other hand, viruses are not living because they cannot multiply independently. They have no cell. So they are acellular. They can perform no metabolism to generate energy. They cannot reproduce independently. They rely on a host. So when you think about virus, we can't say they are dead, neither are they alive. But we can actually consider viruses to be active or inactive. Viruses are active when they are inside a host cell. Now, when viruses are extracellular, where they are not in a viable host cell, then they would have to be in the inactive form. So we can't really classify virus as dead or alive, but we can classify them as active or inactive. And by this classification, viruses can be tend to be said to be obligate intracellular parasites, meaning they need a viable host to be able to function. They need a viable host to perform the function that normal living things go through. So again, viruses, they can be active or inactive, and they are obligate intracellular parasite. They must have a host. They need a host. Now, if you look at any viruses that cause any viral infection, they have their own unique characteristics, which allows us to identify them. Now, some of the unique characteristics is that viruses are not cells, so they are acellular. They are acellular, so they do not have cell. When you think of bacteria, they have prokaryotic cell. Humans have eukaryotic cell, but viruses are not cell. They do not have cells, so they are acellular or non-cellular. They need a viable host, so that's how come they are obligate intracellular parasites. They need a host to be able to survive and function. Now, they also contain genetic material, DNA or RNA. If you look at viruses, they contain genetic material. It can be a DNA virus or it can be an RNA virus. They can have both. Now, viruses also they lack ribosomes because they are acellular. They don't have cellular structures. They lack the ability to go through metabolism. So they rely on the host to be able to produce and generate the energy. They can only reproduce inside a host cell. So they cannot go through replication or multiplication independently by themselves. They need a host to help them. They are everywhere. Now, they are also extremely small in size. They are ultra microscopic. So in terms of size, eukaryotic cells are much bigger in size. And then you have bacterial cells. But comparing viruses to bacterial cells, viruses are ultra smaller, microscopic in size. Between 20 nanometers to 1,500 nanometers in size. They are very, very small when you think about this. But these properties allows us to be able to identify viruses, study them, get to know their behavior, their structure. And this helps us in terms of how we are able to design medication, think of treatment, understand viral infection much in detail. So let's look in detail at some general characteristics. Let's look at some of them in much more details. So first of all, viruses, in terms of their size, they are much smaller than bacteria. And when you compare various viruses that are out there, the largest one we could probably think of as the Pandora virus. And that is about 1,000 nanometers in size. So that is like a giant virus. There's also the herpes virus, which causes the sexually transmitted infection. So herpes is much bigger in size, it's about 150 nanometers in size. Rabies, which mostly animals like dogs, they can be infected by rabies virus. That is also much smaller, is about 125 nanometers in size. The HIV virus is about 110 nanometers in size. Flu, which is a seasonal disease and is caused by the influenza virus, flu is about 100 nanometers in size. Now, when you look at various viruses that we have on the chart here, it clearly tells us that viruses are ultra microscopic. There's size variation among them, and they are too small to be visualized with the naked eye. So similar to viruses compared to bacteria, you need the help of microscope to be able to visualize viruses. And especially for viruses, we use the specialized microscope called electron microscope. Not until 1932, where electron microscope were invented, we could not see viruses. So remember the historical context about microbiology, Antony van Leeuwenhoek and his animal study. Viruses were not part of that category because he was just using a regular lens, compound microscope setup lens to visualize those microbes. Now, with the invention of electron microscope, we're able to visualize viruses at their ultra microscopic level. Now, aside the size of viruses, there are other general characteristics that also help us to be able to identify viruses. Another of such characteristics is the viral composition. Now, every virus have two simplest composition. They're going to have a genetic material, which is either going to be a DNA or an RNA, but you're not going to have both. So no virus have both DNA or an RNA. It only have one of these options. And a Virus would also have a protein code, which normally protects the genetic material. So right here in this image, you can see that this virus has its DNA or RNA in its central core and is being surrounded by the protein codes, also called capsid right here. So these are the two basic shapes composition that viruses do have. Every virus will typically have this composition, genetic material and protein code, which is also called a capsid. Now, there are some details that we want to add to this. So if you look at the protein codes of a virus, it is called a capsid. And this capsid surrounds the nucleic acid, either DNA or RNA. Now, when you combine the capsid surrounding the nucleic acid, it is called a nucleocapsid. So basically, a virus in its simplest composition is a nucleocapsid. Now, in addition to the nucleocapsid, some viruses can have an envelope, and this is a modification of the host cell membrane. Remember, viruses do not have cell. So anything they do have as a feature is coming from their host cell. Envelopes are not found in all viruses. So some viruses do have envelopes, and these are called enveloped viruses. Now, viruses that lack or do not have an envelope are called naked viruses. So that means these viruses only exist as nucleocapsid, where they are only composed of the capsid protein shell and the nucleic acid. But if you are an envelope virus, it means you are going to have your nucleic acid, you're going to have the protein coat, which is the capsid, and you're going to have another envelope, which is shielding around your capsid as well. So these are some basic viral components that helps us in identification of viruses. Now, some viruses may also have spikes on the external of the envelopes. Now, these both naked and envelope viruses can have spikes. The spikes are going to be used as a docking station for the virus on the host cell. And spikes allows the virus to attach really well on the host cell. Now, if a bacteria wants to attach to a host surface, we talked about the fact that bacteria can use fimbriae for attachment. But in this case, viruses have to attach using spikes. And there are various viral types and different spikes proteins that they use. So we'll talk more in details about that. Now, if you have a virus with all these structural composition in place, their nucleic acid, whether DNA or RNA, surrounded by the capsid, and an envelope, whether they are envelope viruses. In this case, for naked viruses, they will not have an envelope. And in both cases, whether naked virus or envelope virus, they may have spikes for attachment. Now, these fully formed virus with all these structural components is called a viron. And the viron is this fully established state where the virus can cause infection in the host cell. Now, if you have the virus and it has all these detailed composition, it is going to be in an infectious viron state to cause an infection. Now, just a little bit detailed into the viral capsid. So the protein coat that surrounds the nucleic acid called capsid is made up of protein subunit called capsomeres. Now, this is very important because the capsid allow viruses to have three different basic structural shapes. Now, if the capsomeres, first of all, are arranged in a rod shape like as a continuous helix, that gives the virus a helical capsid shape. So it's like a hollow tube, continuous helix. That is going to be a helical capsid. Now, sometimes the capsomere can create itself in a three-dimensional shape where it rearranges itself in 20-sided figure with 12 evenly spaced corners. Now, in this case, the viral shape is going to be an icosahedral capsid. Now, in the case where you cannot put the virus as having a rod shape or this three-dimensional shape, then the virus will then be classified as a complex virus. So here is the case where you have tobacco mosaic virus, even the flu virus, their capsomeres, which is the protein code, is arranged in a rod shape as shown in this image right here, where it looks like a helix cylindrical shape right here, like a tube. So rabies virus, which attacks animals like dogs, it also have this helical shape structure as well. Now, in terms of the three-dimensional 20-sided 12 corners shape, you have different dimensions. So, for example, adenovirus, it can have as many as 252 arranged capsomeres. Poliovirus has like 32 capsomeres arrangement. Hepatitis B virus also have a three-dimensional shape, and the same thing for the herpes virus. Now remember, if you cannot put a virus under the helical shape or under the icosahedral shape, then it's likely going to be a complex virus. And a typical example of a complex virus is bacteriophage. It can be a T2 or a T4 bacteriophage. Now, these shapes are typically not symmetrical. So you can't say they are rod shaped, neither can you give it a three-dimensional shape. So in this case, they're going to be classified as complex viruses. Now, if you look at a virus also in detail, we said viruses, some of them may have an envelope, which they are likely going to get from the host cell. Now, the envelope is coming from the cell membrane or the nuclear membrane, and viruses can also have spikes, which they're going to use for attachment. And these spikes are typically glycoproteins that are going to be essential for the virus to attach to its host of concern. So just a quick summary. Viruses do have genetic material. And this genetic material can come in the form of DNA or RNA, but not both. Now, viruses, in terms of the entire genome or their genetic material, there's variation in it. Some of it may be large, some of it may be small. So, for example, hepatitis B only has 4 genes in its genome. The Pandora virus, which is one of the giant viruses in terms of size, they do have over 250,000 genes. So there's definitely variation in the number of genome, which is the entire complement of DNA or RNA that a virus can carry in itself. Now, when you look at this genetic makeup, whether DNA or RNA, this is very important because it is one of the main things that allows us to classify viruses in the gray area of whether they are alive or not. Because with their genes or genetic makeup, they can invade a host cell and they can use their genetic or genes to redirect the activities of the host cell. So the genome, which is the genetic material, is really, really important for the virus because it allows us to take over the host cell. Now, in terms of the DNA or RNA, for RNA especially, you can have positive sense RNA viruses and you can have negative sense RNA. Just a quick recap, RNAs are single-stranded compared to DNA, which is double-stranded. So in the case where viruses are RNA viruses, it's going to be single-stranded, but some have a rare case where we do have double-stranded RNA, and we would highlight on that shortly. But in terms of the RNA, positive sense RNA, these are single-stranded RNA genome, which can easily be translated into proteins. Remember, the genetic information flow is from DNA to RNA to proteins. So even for RNA viruses, we still want to be able to translate from RNA to proteins, which is for regulation or for function. So positive sense RNA viruses is such that their genome is ready to be translated to create proteins. On the other hand, viruses which have negative sense genome, you need to convert them to the proper messenger RNA or mRNA form before they can now be ready to be translated into proteins. So we'll take a look at a little bit of details into how these genetic or nucleic acid information really works because it is key for the viral activity. Now, first of all, we can classify the genome or the genetic material of viruses into four key categories. We can have double-stranded DNA. So DS stands for double-stranded DNA. Now, typical examples, so herpes virus that causes herpes, it has a double-stranded DNA. Now, DNA is typically double-stranded, where you see the double helix, right? So it's typically double-stranded, where it has two strands. Now, RNA, on the other hand, is single-stranded. But in this case, you have a double-stranded RNA. Now, for double-stranded RNA, the strategy is to be able to convert them to messenger RNA before they will be ready for you to translate to give you proteins. So we have more regular double-stranded DNA. There's regular single-stranded RNA, which can be positive or negative, but you rarely have single-stranded RNA and double-stranded RNA molecules in viruses, but they do exist. Now, if you look at viruses, these are all examples of viruses and the type of genome that they have. So the rotavirus, which normally causes gastroenteritis, it has a double-stranded RNA. Poliovirus has a positive sense single-stranded RNA, which means you can easily translate it to proteins. In this case, the parvovirus, which can cause skin rashes, it has a single-stranded DNA molecule. Now, this genome classification, as observed in these different viral types, allows us to know how to understand how the virus can attack the host cell and use the host cell machinery to take over the activity of the host, multiply itself, and take over the host cell. Now, HIV, on the other hand, is a single-stranded RNA, but is an RT, which stands for retrovirus, Now, for a retrovirus, we need to revert it back to DNA. So it's an RNA virus, but for it to function properly, the retrovirus allows us to reverse transcribe the RNA back to DNA and then back to RNA again before we can get the RNA being converted to proteins or being translated into proteins properly. So retrovirus is very unique for HIV virus. Now, flu, which is very common, is a single-stranded RNA, but is a negative sense, which means you need to be able to convert it before you can convert it to a good translated protein version. So remember, negative sense is not ready for you to translate it, but positive sense means it's ready for you to translate it in this particular case. Now, for viruses, they are able to take over host cell, they can multiply, and they can cause so many diseases to us. But we first want to understand viruses based on how they are classified and named, and we'll use that information to help us understand how viruses multiply. Now, in terms of viruses, the International Committee on Taxonomy of Viruses, the ICTV, is the recognized scientific body responsible for organizing, for naming viruses. Now, unlike the other classification that we've looked at, where we started from the domain all the way to the species level, viruses are named mainly from the order level down to the species level. Now, the order level, the name ends with viralis. So anytime you see the word viralis ending a viral naming, it clearly shows you that is an order level of classification. Viridae means is a family level classification. So Coronaviridae, that means we are talking about the coronavirus family virus classification. Now, in terms of how we can classify virus, we typically also use their genetic information to classify virus. So viruses can be classified based on the fact that they are DNA virus or they are RNA virus. So for example, herpes viridae, which causes herpes. Normally for such disease, you'll be able to see there's fever, there's sore, there's blisters, as shown in this image right here. So So they all belong to the herpes viridae family, and their genetic makeup or their genome is DNA in this case. Now, you also have the papilloma viridae or the papilloma virus, and a typical example is the human papilloma virus, HPV, which causes several wat types that we see. Could be on the legs, it could be on the hands, or any part of the body. Now, these are also DNA viruses, and the viridae clearly tells us that we are dealing with viral families. Now, in terms of RNA virus, some common example, you have the flaviviridae, where you have the flavivirus or the West Nile fever virus, which is commonly known. Now, this causes the wet Nile disease or wet Nile fever. And this is one of the important disease which is caused by a virus, because this virus is carried by a simple mosquito bite. The virus is transmitted through the simple mosquito bite that can affect your brain. And when you look at this virus in the Flaviviridae family, this is in the same group as the dengue fever, also as the yellow fever, where some of the typical symptoms that is observed is like fever, headaches, or rashes. Now, 80% of people who are exposed to the West Nile fever, they don't really show symptoms, but in the cases of where symptoms are observed, you will see rashes on the skin. So this is Flaviviridae, where the West Nile virus is also part. Now, we all live through the pandemic, the COVID-19. It belongs to the coronaviridae virus. So the SARS-CoV-2 virus, which causes COVID-19, is an RNA virus. So anytime you've heard of mRNA testing, PCR testing, you are actually dealing with the RNA of the COVID virus because it's an RNA virus. Now, HIV, as we mentioned it, HIV is a retroviridae virus. Because remember, we said it has a retro virus because you need to reverse transcribe its RNA back to DNA before you can fully convert it to a proper translated protein. So HIV belongs to the retroviridae family. So now that we know a little bit about viral classification, we've all come in contact with viruses. How do viruses multiply to take over the body of a host or take over the body of their viable host cell? Now, viruses uses 6 key steps through these phases, typically for animal viruses, where they use this to multiply in their numbers. Now, the very first step is adsorption, also known as attachment. So before a virus can attach and multiply itself, it needs a viable host. And when it finds a viable host, it is going to attach. And viruses are going to use their spikes for the attachment. Now, if a virus managed to attach, it now has to go through the second phase where it will penetrate the host cell. It will penetrate the host cell. And then after penetration, it will uncoat itself to release its genomic material. So if it's a DNA virus, it's going to uncoat to release its DNA. If it's an RNA virus, it's going to uncoat and release its RNA. And then the next stage will be synthesis. The virus will now use the host cell to synthesize more copies of its DNA, or if it's an RNA virus, to synthesize more copies of its RNA. It would reassemble its parts together, and then it will be ready to be released from the host cell. So viruses go through these 6 key cycles to be able to multiply in their numbers in the host cell and take over the activities inside of the host cell. So let's take each phases one after the other. So the first is adsorption, where the virus would attach and have an encounter with the host cell. Now, viruses have spikes. So right here in this image, you have an envelope virus with its spikes, and it's going to use the spikes to attach to a host cell. Now, viruses are going to attach based on specific receptor sites on the cell membrane of their host. So there are various viral types, and they bind to specific receptors on specific cell types. So, for example, if you have hepatitis B virus, where they typically infect liver cells of humans, they are restricted hosts, which means they bind and they have specific receptors on human liver cells where they can really be compatible to bind. Now, you can also have some viruses which are broad hosts, and a typical example is the rabies virus. So, for this type of virus, they can infect various cell types, which means they can attach to various receptors on the surface of different cell types. Now, an HIV virus or a SARS-CoV-2 virus, all these are different cell types. So, in the case of SARS-CoV-2, they bind to the ACS2 receptors on the cell surface. So clearly, viruses do have specific receptors on host cell where they can bind. Now, there are some even viruses that are also very restrictive. So, for example, HIV virus. If you take the HIV virus, it doesn't bind to just any cell type surface. And even cell type, they are even more specific in where they can bind to the receptors. So HIV binds to CD4 receptors on T cells of our immune cells. So they are very, very specific in the type of receptors where they can even bind to on the host cell. So this is really critical because when we are designing intervention or we are designing different ways to understand viruses for treatment purposes, we can clearly use how they attach to the host as a good mechanism to understand how to come up with strategies to treat viral infection. Because remember, viruses need to first attach to a host cell first. Now, when they attach, they're going to penetrate using a process called endocytosis, or they can use membrane fusion. Now, endocytosis would allow an entire virus to be engulfed by the host cell, using vacuoles or vesicles to be able to enter. Now, when the virus is able to penetrate, they would uncoat themselves where the capsid is removed, the capsid, which is the protein coat. And when they uncoat themselves, then now you can have the genome being fully released. So if it's a DNA virus, the DNA would be released. If it's an RNA virus, the RNA would be released in this case. Now, during the synthesis stage, the virus is going to use the host machinery to take control and multiply its genomic material. So RNA viruses are going to multiply and replicate inside the cytoplasm, but DNA viruses are going to replicate and multiply inside of the nucleus, where they can make more copies of themselves. Now, after they've had more copies of themselves being made, new virions would be assembled and they will be ready to be released. Now, the release out of the host cell can be through budding or exocytosis. Now, in the case of non-enveloped viruses, viruses that do not have an envelope, they are going to use merely cell lysis. They're going to use the process of cell lysis to be able to get themselves out of the host cell. Now, in the process of cell lysis, they can release an enzyme called lysozyme to be able to break the host cell membrane so that they can have an exit out of the cell. So they can use lysozyme enzyme to be able to digest the host cell membrane and then lysis themselves out. Now, envelope viruses cannot use cell lysis method because the lysozyme can mess up with their envelope whilst they are being released. So they prefer to use budding, where they can be pinched off, and their pinching off would allow their entire structure together with the envelope to be released intact. And they can also use exocytosis, where their entire structure is pinched off and exported outside of the cell. So in the case of envelope viruses, their release has to be based on budding or exocytosis in this case. Now, this is a very nice cycle for viruses. And typically, if you have viruses, one virus which enter the host cell, depending on the type of viruses, you can have several varying numbers of virons which are being released. Now, for an example like pos virus. So for pos virus, if you have one virus which make entry into a host cell, about 10,000 virons can be produced, assembled, and released out of the host cell. Now, in the case of poliovirus, from going through all the 6 cycles of phases, you can have about 100,000 virons per cell being released out to infect new host cell in there. Now, there might be several variations depending on the cell type, but the key thing to note is that one virus can start through the multiplication cycle, but can end up having either 1,000, 10,000, or 100,000, or even more being released from the cell. And it's going to be basically based on the type of virus in question. Now, there are some viruses that do not go through this cycle in its entire phase in a complete set. There are some viruses that they harbor inside the host cell and do not lysis immediately. Now, these are called proviruses. They have persistent infection. These viral types have that kind of infection persistently because they attach themselves to the host, they release their genetic information in the host, and instead of synthesizing and destroying the host cell, they incorporate their viral DNA into the host DNA. And this creates what we call a chronic latent state, where during this period, it's going to be a period of inactivity. Now, during this period of inactivity, the virus will be inactive, but whilst the host cell multiplied, the virus also multiplied alongside it. Now, aside these viruses, which are provirus, we also have oncoviruses. So oncology, the study of cancer. So oncoviruses are viruses that cause cancer. Now, 13% of human cancers have been linked to viruses. And there are various viruses that have been connected. So, for example, cervical cancer has been connected to the HPV or the human papilloma virus in this case. Now, if you look at oncoviruses, the viruses are able to cause cancer. It can be directly or indirectly through the process of transformation. Now, the virus can carry genes that can directly cause cancer, or the virus can produce certain types of protein that can trigger loss of growth regulation inside specific cell types. So the virus can release certain proteins which allow certain cell type to grow and proliferate profusely without being able to stop. Now let's look at some typical example. So cervical cancer, like I mentioned, have been linked to the HPV. The herpes virus is also connected to Burkitt lymphoma. But I want you to do a quick research for me. Hepatitis B virus, what type of cancer is connected or linked to the hepatitis B virus? What type of cancer is being linked to the hepatitis B virus? Please share with me your answers in the comment section right here. Now let's continue. So in terms of virus, there are also viruses that can infect bacteria. Remember in the background information, I told you that viruses are abundant and they can have virtually every cell type as their host. They can infect plants, bacteria, humans. Viruses that infect bacteria are called bacteriophage. And these were discovered in the 1915s, and most bacteriophage were identified to contain double-stranded DNA molecule structure. But you can also have some that exist as single-stranded RNA or double-stranded RNA. These are known to infect bacterial species. They can parasitize bacteria as their host and take over the bacteria machinery to multiply or replicate themselves. Now, oftentimes, if a virus infects a bacteria, they make the bacteria more pathogenic, which means they can cause more harm in humans. So the bacteria become more pathogenic for humans, where they can make the human more sick than they usually could be. Now, two typical examples of bacteriophage. We'll just look at one, which is the T2 bacteriophage. This virus infects Escherichia coli, also popularly known as E. coli. Now, they might sometimes go through the similar same cycle that we've talked about in terms of how they multiply, as we discuss in animal viruses, where they can absorb, they can penetrate, they can uncoat, they can synthesize, they can assemble themselves, and then they can release. But more specifically, they go through two main specific cycles, which is the lytic cycle and the lysogenic cycle. Now, the key thing about the T2 bacteriophage in this cycle is that how it handles its host in each of these cycles is a little bit different. Now, if a bacteriophage is going through the lytic cycle, it will go through the 6 stages that we talked about, and it will destroy its host cell immediately after release. Now, in the case of lysogenic cycle, the virus doesn't destroy its host cell immediately. It takes time before it does that. So let's look at this two in much more detail. During the lytic cycle of multiplication of a bacteriophage, it is going to go through the same process. So the bacteriophage is going to attach itself to the E. coli host right here and is using its tail spikes to insert itself into the E. coli host right here. After attachment, is going to now penetrate the E. coli host. It is going to uncoat itself to release its genomic material, use the E. coli machinery to synthesize more copies of itself, assemble, mature, and then it will be ready to release. But before it is released, it's going to lysis the host cell using lysozyme that we've talked about. And this is going to weaken the host cell completely. And then now, upon its release, the host cell is going to be lysis and is going to die from that. On the other hand, a virus going through the lysogenic cycle, the virus is going to go through the attachment and the penetration stage. But instead of using the host machinery to synthesize more copies, the virus is going to incorporate its DNA into the host DNA. And they're going to create what we call a prophage. So the prophage is when you have the viral DNA incorporated into the host DNA. Now, during the lysogenic stage, where the viral DNA is inserted, anytime the host multiplies, the virus genome is also multiplied in the process. So it would keep multiplying, until this induction stage. And during the induction stage, the virus would now detach itself from the host DNA. So again, just to reiterate, during the lysogenic stage, the virus is going to insert its DNA to create a prophage. So this is going to be an inactive state. And when they are active, they're going to go through a process called induction, where the prophage would detach itself. The viral DNA is going to detach itself from the host DNA. And then now the viral DNA is going to go through the lytic cycle again, where they would now use the host machinery to make more copies of themselves and then begin to synthesize, assemble, and then finally get released. Now, typical example of viruses that go through this lysogenic stage is the virus that cause diphtheria, botulism, and even cholera toxins in this case, where they can be latent, they can be inactive for a long while, but they will still definitely show up and go through the lytic cycle where they would eventually kill the host cell in question. So just a quick recap. If a bacteriophage is going through the lytic cycle, they will destroy the bacterial cell immediately. But if they are going through the lysogenic cycle, the bacteriophage is going to become incorporated into the host cell, where they can stay there inactive as a prophage for a long time until they are induced to become active again, and then they can cause infection to the host cell and completely destroy the host cell upon lysis. Now, viruses, as I said, they are invisible invaders. They cause so many diseases to us. But typically, we do not have medications for viruses that could potentially not cause any harm to human cells. So to be able to understand viruses, there has to be a way to culture them, grow them, and then this can be used as models for us to study different medication and treatment options for them. So being able to cultivate viruses is very important because it allows us to identify them. It allows us to prepare vaccines for them because viruses are acellular. So normally antiviral drug medication design or development is very challenging. So one of the effective means towards viral treatment is creating vaccines. And since viruses need viable hosts, we need to be able to learn how to culture and grow viruses to help us in creating vaccines. Now, when we culture viruses also, it gives us a good detailed research into their structure, how they multiply, what are their genetics, how do they infect different host cells. Now, to culture viruses, we can use in vivo methods where we have a living organism model, which we can use. So, lab animals, embryonic bird tissues could be used. We could also use in vitro, where they are done with cell culture or tissue culture conditions to be able to study viruses. Now, in case of in vivo animal models, we can use animals like mice, hamsters, rats. We can inject the skin, foot pads, muscles, blood, just to be able to get a good understanding and use these host sources to help the virus to replicate, to multiply. Now, we can also use bird embryo, in this case, which is an egg. It is one of the most useful models because, for example, the bird embryo, the yolk gives it a sterile environment and it also have nourishment in it, which can also help the virus to be able to function really well. In terms of tissue culture, you can use various cells. So, for example, for viruses, a typical example is you can use bacteria as a host to have like a plague assay where the bacteria are growing. You can now add the virus to it to be a viable host for the virus. Now, viruses are very important. So, just like we talked about bacteria, there are so many health diseases that are associated with viruses. So it's very important for us to measure and understand viruses. But the infection of virus is worldwide, is global. There are various infection, whether it's acute or chronic, which can be related to virus. So common cold, chickenpox, the flu or influenza, herpes, and what, all these are caused by viruses. Now, some viruses even have high concerns when it comes to health because they can even have as high as 100% mortality. In the case of rabies, there's 100% mortality. Ebola had between 25 to 90% of mortality rate, depending on the region that the data was reported for that. Now, there are some viral infection that can even lead to long-term disability or long-term deformity, such as polio and neonatal rubella. That is why sometimes there are vaccines for some of these viral infectious diseases out there, just to be on the preventive side. So again, for viruses, one of the important therapeutic for viruses is vaccines. And viruses are able to mutate at a quicker rate. So if you think about it, the flu shot you got for last year is not the same flu shot that you got for this year. And that is mainly because viruses are constantly mutating at a raster pace, very fast pace. So even though vaccines are good measures, as good therapies, there have to be evolving vaccines to be able to match up this quickly mutating viral state. So there are also some antiviral medications that have been developed because antibiotics are typically not effective against viruses. There's been interferons, and this we will talk much in details when we start talking about the immune system and how our body fights its own infection. So we'll talk about this particular cytokines that are in there. But the main goal for viral infection is prevention because antibiotics are ineffective. So most of these antiviral drugs, they would be seeking to make sure that the virus do not attach to a host cell or the virus not able to penetrate to a host cell. But for viruses, being able to have vaccines as therapy for the immune system to build and fight against this is one of the most advantageous therapies which is out there. Now, in addition to viruses that we've talked extensively about, there are other non-cellular infectious agents, and one of them is prions. So if you've heard of mad cow disease, yes, they are not caused by viruses, but they are caused by misfolded proteins, which are called prions. Now, prions are not DNA, prions are not RNA. Prions are simply proteins that are misfolded. Now, these misfolded proteins can cause our normal proteins to also misfold. Now, if you've heard of mad cow disease or brain encephalopathy, spongiform encephalopathies, these are all caused by prions. So prions can cause misfolding in the brain and begin to create sponges on the brain, where the brain is not able to function properly. Now, another viral particle that I want us to talk about lastly is viroids. So these are not viruses, they are virus-like agents, but they typically attack plants. They are very small in size. They are about 1/10 in size of a typical viral shape that you would have. Now, these viroids, which attack plants, they are simply single-stranded RNA. They do not have capsid like regular viruses have. They only exist as stranded RNA molecule. And they can attack plants like tomatoes, potatoes, chrysanthemums, and several other plant species which is out there. So viruses are typically, we've seen them where they have their capsid and their DNA, but viroids, these are small RNA molecules and they only infect plants. Now we can also have what we call satellite viruses. So this satellite viruses requires the help of other viruses so they can infect other viruses to be able to thrive and also cause infection in the body. So if you look at everything that we've talked about in this chapter, viruses are very invisible, but they are mighty and powerful in how they attack the body. And it's important for us to understand their structure, learn about their genetics, know about their shape, get to know their structural cellular details and how, even though they are obligate acellular parasite, they do not have cell, it's important for us to understand how they attack a host cell and how they are able to cause various diseases that we are exposed to or we are experiencing on a daily basis, like the flu, common sore throat, COVID, liver diseases. And now we even know that there are various cancers that are even related or are caused by viruses, like cervical cancer, liver cancer, and several other cancer types. Now, of course, we've also highlighted the fact that we don't know whether they are dead or alive, but we do know that they can be active or inactive. We do know about their structural composition. We do know about their genomic type. We do know about a few basic details about viruses. But one thing we do know for sure about viruses is the fact that their treatments, when they cause diseases, is mainly through antiviral drugs or vaccines. But vaccines are noted to be one of the most very highly powerful therapeutic control for viral infection. So viruses, they are simple in structure, but they are powerful in their impact. They need a host cell to be able to evolve and multiply rapidly, and they can cause widespread diseases, just like we experienced for the COVID. Now, there are other viral particles such as prions, and prions reminds us that there can be proteins which can equally be infectious. So in this lecture, I hope that you understand viruses and prions, and you can go back and review portions of it to understand different sections that we've had. But this summary chart clearly put it in perspective that similar to bacteria, similar to the other microbial organisms that we've talked about, viruses are simple in structure. We can really get to know them in much detail. We can understand how they work, and we can get to see how we can come up with treatments and vaccines and other control strategies to treat viral infections that may come up. Feel free to let me know if you have any questions, and I'll be glad to answer you. My name is Dr. S, and this is Biology with Dr. S. And let me know any thoughts or questions that you have in the comment section. Thank you.